So myself, Mohan Kumar TY, and I'm going to give my presentation regarding one of the German scientists who called Paul Enrich. So next slide, sir. Okay. So before getting into the topic, we will have an introduction about the scientist who we called Paul Enrich. He is one of the German physician and the scientist who worked in the field of so many branches of the biology that are called to be the physiology and the medicine. And he got a Nobel Prize in the year 1908, and he is considered to be the modern father of chemotherapy, and he founder of chemotherapy. And next, he is having a wide contribution in the field of histology, that means the study of tissue and microscopic cells, and hematology, a study of blood organs and branches which deals with the study of blood, immunology, immune system, oncology, study of cancer, and microbiology and pharmacology. And next will be he invented standardization of manufacturing the anti-diphtheria serum and synthesis of antibacterial drug, and the concept of chemoreceptor and chemotherapy, and the concept of developing the resistance. So next slide, sir. So next slide. Allow. No, sir. Previous one, sir.
Previous one.
Sir. Okay. He born in the year 1854, March 14th, and he was called to be the microbe hunter, and he cartoon as a Dr. Fantasist like a god, and known as the father of chemotherapy. Why? Because he uses the chemical substances that are, especially those are produced synthetically, that are used to destroy the pathogenic microorganisms within the living cells or a body. And he is considered to be one of the revolutionists interested and histological style. Next slide, sir. When we talk about this revolutionary introduction, means he studied about the WBC white blood cells that can be stained using methylene blue stain. Next slide, sir. And he also brings about the standardization of manufacturing the anti-diphtheria serum, and he brings about the drug to cure the disease called to be the diphtheria. Next slide, sir. Okay. So when we talk about his career, during his career, he exploited his knowledge of chemistry, and thereby he was able to merge the cellular and molecular theories into a new concept. And subsequently, he defined the biological principles and demonstrated their practical applications and applicability of the medicine. By doing so, Paul Enrich established the basic principles of translational medicine. That means the evidence researchers or the disease researchers between the human cells and the medicines. And these principles and hypotheses not only become tremendously useful, but many of its ideas
Not audible. Not audible.
side chain theory, which is hypothesized by the Paul Enrich, and he stated that he worked on an antitoxin and the diphtheria and their bindings to the antibodies in the blood. Hypothesized that antibodies that can bind to the antigen through a special chemical structure, and he termed it as a side chain, which are later termed as a receptor. So these receptors will have a membrane-related structure that can interact with the extracellular molecules that are called to be the antigens or the ligands. And this theory was later extended to a generally applicable receptor-ligand concept, and that greatly influenced in the field of physiology, immunology, hematology, and pharmacology, and still fundamental in science today. The next slide, sir. And he concluded that there will be a strong affinity must exist between the biological structure and the strain who is applied. That means he studied the entire spectrum of the staining technique and chemistry which is involved in it, and differential staining of tissue using a methylene blue. That means more than one day he used it to differentiate between different types of tissue or the cell. Next, he detect the bilirubin in the urine of a patient who suffers from the disease called a jaundice, and he stained and named the mast cells that are parts of the immune system using an alkaline dye. These are oval in shape or irregular in shape and having a nucleus and other organelles in it. And he differentiated the leukocyte, that means a part of immune system, into a basophils, eosinophils, and neutrophils. And first he showed the nucleated RBC, and he subdivided the RBCs into megaloblast, microblast, normaloblast, and poikiloblast. Next, methylene blue also stained the long appendages of nerve cells, and he developed a new staining procedure for mycobacterium, and which later became the basis of current zeal-nalcin acid fast staining. So next slide, sir. Now is research on serum. He formulated the side chain theory as we discussed earlier, proposing a possible explanation in the process of immunity. And he gave a concept of acquired immunity and the mechanism of transferring the immunity from mother to the fetus. And along with he standardization of antidiphtheриal serum production process. So next slide, sir. And he also worked on the research of oncology, that means the study of cancer. And he achieved insight that when the tumor cells are cultivated by transplanting the tumor cells, their malignancy or the tendency increases from generation. And if primarily tumor is removed, then metastasis precipitously increases. And the magic bullet about the Paul Enrich, if a compound could be made that selectively targeted a disease-causing organism, then a toxin for that organism could be delivered along with the agent of selectivity. And need to study the relationship between the chemical composition of the drugs and the mode of action in the organism. And selective drugs were needed which, like antitoxin, aimed specifically at their corresponding toxins. And high parasitotrophism with low organotrophism. These are the magic bullets about Paul Enrich. And this is about my topic.